My name is Sue Abernathy, and I'm doing my project as a research proposal. So the title of my project is Phytoremediation Potential in Hemp, and I'm specifically looking at the cannabinoids and how they might change under toxic growing conditions. Okay, so a little background about hemp. So hemp is Cannabis sativa L. There's also Cannabis indica and Cannabis ruderalis. Sativa is typically grown for fiber, whereas indica is typically grown for the drug version, and the ruderalis is the wild type. So hemp is in the Cannabaceae family. It's herbaceous C3 annual. Its ploidy is 2n equals 20, so there's 10 chromosomes, 2 copies of each. It's dioecious naturally, although it can be grown monoecious, especially with feminized seed. Hemp is a short day plant. It needs 10 to 12 hours of darkness for hemp varieties and 13 to 14 hours for marijuana varieties to induce flowering. Hemp versus marijuana, biologically, they're essentially the same. However, legally, hemp is defined as having 0.3% THC or less. Hemp can be grown spaced out or in very dense stands. If it's dense, typically grown for the fiber, it can grow up to about 16 feet in height, where you have reduced branching to produce the most fiber in the stem. Then you can have spaced out kind of hemp where it grows shorter, more like 3 feet, because it puts more biomass into branching. And this works well for the chemical compounds in the flowers. And then for seed varieties, it's kind of in the middle. You want to maximize as much of each quality as you want between seed and fiber. So these tend to be a little bit shorter than typical fiber, but most of the flowers are at the top where they collect the seeds. So hemp has 50,000 uses, and these come from the stem, seeds, and flowers. Okay, and so moving on to hemp's chemicals. Hemp does produce many chemical compounds. These are in the glandular hair-like trichomes, which are multicellular. The trichomes, the compounds in the trichomes are secondary compounds, so rather than being used for reproduction or structural biomass, these are usually used for things like protection from herbivores and pathogens. Hemp chemicals, hemp cannabinoids, are unique trichome synthesis in the fact that they're made in the trichomes, these different compounds, rather than being made like in something like tobacco, where it's made in the root and then transported to the trichomes. The cannabinoids are the most dense in the trichomes of unpollinated females, and they're also responsible for other things like the flavors, fragrances, pharmaceuticals, therapeutics. So why does studying hemp's biology matter? Because hemp is very recognizable. It's super controversial with marijuana. However, it's very biologically unknown. There's very little that's known about it. So understanding hemp's biology leads to understanding hemp's potential. And this could be potential in many different areas. So again, why do we want to study hemp? Here's another reason. This is from the Small Book, 2017. And this shows hemp's friendliness. So on this chart here, or this graph, on the right side in the green is environmentally friendly. So these are the positive values. And on the left side, the reds here are negatively or not as environmentally friendly. And so the yellows highlight the 3 different forms of hemp. So we have fiber, oilseed, and the drug cannabis. And both fiber and oilseed show on the positive side, and the drug is on the negative side. And this, they think, is mostly due to irresponsible growing practices. So this was done, they figured the ecological friendliness with 24 different criteria. And these were things like how much water, herbicide, pesticide, fungicide, fertilizer, and fossil fuel use, whether there's polluted soil, water, and atmosphere created from hemp? And then is there displacement or destruction of native plants and animals and microorganisms? So also hemp is beneficial when added to other crops that are already growing because it can increase the profit, it provides a new market, and it cushions failure of the traditional or original crop that was growing, and it provides fewer pests than a typical monoculture would provide, and creates wildlife niches. So if we're looking at the background of the cannabinoids, a little bit about their anatomy. So the trichomes are found in these bracts, which is sort of like a modified leaf, a single leaflet, and that's where they are the most dense. This is around the ovary, and here you have the stigma. And so this is called a perigonal bract. Sorry about that. So the trichomes are found in these bracts, but they're also found in leaves, stems, and actually even on anthers. So this is a close-up of a trichome right here. And here's a bunch of trichomes on a young leaf. And you can see there's different sizes. So they actually do come in sessile, where there's really no stalk part right here, versus a medium or a long one, where there's a whole bunch of, these are epidermal cells that rise up. So the top of it right here has these disc cells, and this is actually where the compounds are synthesized. So remember, like I said before, they're actually synthesized here in the trichomes rather than transported here. And then they're stored in this top part here, which is really turns into like a sticky resin, where it has the different cannabinoids and terpenoids in it. So this will be obviously more CBD if it's grown for fiber or for medicinal purposes, and for the drug, this would contain more THC up here. So then looking at the pathways of these cannabinoids, there are two precursor pathways, GPP and OA, that synthesize and make CBG. And then synthase convert this CBG, it's actually CBGA, into, they convert non-enzymatically from the acid form to the neutral forms of each of these, of the neutral forms here. So CBD, actually I think this one is supposed to be CBC, and then THC, CBD. So the CBGA converts the THCA into the THC, for example. So for chemotypes, we have one gene with two codominant alleles. So the B locus alleles, we have BT and BD that are actually controlling the biosynthesis of these compounds. So to get CBD, it's homozygous for BD, BD, and for THC, it would be homozygous for BT, BT. And then for CBC, it would be BC. I think it's BC, BD, if I'm not mistaken. And CBC is actually independent of the others, and it's found in juvenile hemp. And then CBN is found in mature hemp, and it's degenerated from THC. So this middle one shows, here's this acidic form of THC with its carboxyl group. It gets decarboxylated into THC, the normal version, or the neutral form, sorry. And then this, as the plant grows and hemp becomes more mature, it degenerates into CBN. And then the bottom one here is showing you if you have more CBD, this is what the compound looks like, found off in the fiber, oilseed hemp. And then the THC, this is its compound. And here it shows it as marijuana, but if the THC is less than 0.3, this would actually be hemp right here. But if it's higher, it would be marijuana. And then in indica, you tend to have more, or both of them, the CBD and the THC, although you could still have that in the sativa as well. But I just thought that was kind of interesting to show. Okay, so now we go to the background of phytoremediation. So phytoremediation is environmental cleanup using plants. And this could be using accumulators or hyperaccumulators to maximize pollutant extraction. So that means accumulators are just a plant is able to accumulate a certain toxin or element, And a hyperaccumulator is it accumulates much more, like 1,000 times more than normal. And then usually in the hyperaccumulator situation, the plant typically would die. Other close versions of it would, or varieties of it would. So phytoremediation, in addition to cleaning up different toxins, can also be used for biofortification, where it's pulling out different elements that can be used for nutrition and added to other crops. And it also can be used for phytomining. Phytomining is when they would pull up elements, something like nickel, where you would just want to pull it out of the ground. And you can do that with plants, and so it's less damaging than traditional methods of mining. So once these things are, these contaminants are pulled up, they have different fates in the plant. So in the roots area down here, we have phytostabilization. So this is when we have a contaminant that is actually stabilized in the rhizosphere of the roots. So it just holds it and keeps it from leaching somewhere, but it doesn't actually remove it. Then we have phytostimulation, where it breaks it down, and it could stay in the soil then as more as non-toxic forms, or it could be released into the atmosphere here from the soil. Then you have phytoextraction, which is storing it. Usually this happens in the leaf, and it's usually in the vacuole is where the contaminant would be stored. Phytovolatilization would be when you maybe would methylize the toxin and volatilize it out into the atmosphere. Or phytodegradation is when it breaks it down in the leaf or somewhere in the shoot into non-toxic forms. Okay, so phytoremediation then has different phytotechnologies or ways that it can be used. So we have rhizofiltration, which would be floating plants on the surface of water and filtering through their roots to pull out the contaminant. This works really well with something like duckweed, duckweed, where it floats on the surface and has very long roots that can pull out contaminants. You can do that in something that you build, or you could do it like in a river or something, as long as you have flow that would go through the roots. You can also make a constructed wetlands, where sort of the same idea as this right here. However, you would build it, and you could use things like cattails that would actually be in the bottom soil that would again pull out contaminants. This could also be something like a barrier, where you've got water flow going down into a river, and it's got contaminants in it. Let's say maybe from farming runoff or something, and you could have willows growing that would filter that water before it goes down into the river, so it would prevent things like dead zones. Then you can have air filtration, something like a tree, like ginkgos or something that you see a lot of times in cities, where it filters contaminants out of the air. And then hydraulic control, so trees are really good for this, where you have water that's down deep. Trees have very deep roots. You can actually plant trees down lower to get them to reach all the way down into, like, say, a water table or something, where it'll extract contaminants up into the roots. And then you can have field pumps. So this works really well with something like hemp, where if you had a source of water, like say down here, or it could be a river or any kind of like a lake or something, or pond that's got contaminants in it, you could pump them up and out. And then say this was a hemp plant right here, you pump them out and then water the field of hemp. And then as it comes down through, the hemp would then pull the contaminants out of it. So then there's phytoextraction, which is just if you had a field just in the soil, you have your contaminant, and it's just pulled out of the soil into the plant. However, this could also be stabilization, where it just holds it in the roots. And then, so that's and then phytostabilization over here would be preventing leakage down into the water table. So it's going to hold it right here in the root zone, but which is good, but it prevents it from running down, but you're not actually able to pull it out of the ground necessarily. And then again, we have biofortification. This works really well with something like mustard, Brassica gentiana, that's able to phytoremediate selenium, for example. And phytomining, alyssum is real good at pulling out nickel. Okay, so this is kind of a cool chart that kind of tells you whether something's going to work or not. This is from a book called Phyto by Kennan and Kirkwood, and it just gives you a general idea of whether a contaminant is phytoremediatable, I guess you could say. So this is comparing the contaminant potential over time or versus time. So here we have good potential, some potential, and less potential. And this is time across the X. So right here is about 10 years. So this first part would be less than 10 years, and this is beyond 10 years. Is how long that it would take for the cleanup to happen. And then the squares are inorganics and the circles are organics. So right here in the greens, we have inorganics, and they show as a good potential, and they don't take a long time compared to the others. And then in the yellows, we have chlorinated solvents, something like you'd see at dry cleaning places. And then the reds are petroleums, which still do fairly well, but it could take up to 10 years to clean these up. And then some potentials in the middle, we have the oranges are pesticides, and then the purples over here, and these guys down here, these are all metals or metalloids. And then for less potential, either they take too long or they're just too hard to pull out, or there's nothing that will phytoremediate them. We have salts, we have explosives, radionuclides, and we have POPs, persistent organic pollutants. So that kind of gives you an idea of whether something will work. And then these different figures right up here, this key shows you what kind of technology works well. So like the lock is the phytostabilization. The one with the little roots right here is rhizofiltration. These arrows right here are showing volatilization, and then breakdown in the plant like degradation. Okay, so is hemp fairly good for phytoremediation? I think probably it is. It has some properties that make it very viable for phytoremediation. It's very hardy, has deep roots, it's fast growing, high biomass, economic value is good, and it compares well with other phytoremediators like mustard and sunflower. So you need to select the right plant for what you're screening. And in hemp's case, probably something like field extraction with inorganics would be its best choice. So just a couple of things to show hemp, previous studies that hemp has done with some inorganics. So this one's a study by Mears et al. In 2005. And in this one, they did soil amendments, and they added soil amendments to see how well things were pulled out of the ground or accumulated. So we have the letters A through D. So A is hemp, B is sunflower, C is mustard, and D is corn. And then 0 was, they only added water. 1, they added EDTA, and 2, they added EDDS for amendments. So on this one, this is biomass, and it shows right here, these A's are the hemp with the water, with the EDTA and EDDS. And so it produced more biomass than the sunflowers and the mustard, but not quite as much as the corn. So with zinc, this one now they split them up. So the first, this section right here is just with water. These guys have EDTA added, and this has EDDS added to it. So you can see that the hemp, which is the A, did pretty well with the EDTA as well as the EDDS. And that was with zinc. And then with copper, again, it did quite well with the EDDS. And then with nickel, same thing. It did quite well with EDDS, and it did better than the others with EDTA. And then one more study. This one is Stonehouse et al. 2020. And this shows selenium. So this one, they did seedlings up here and mature plants down here. And they found this is biomass, and so it was found that hemp did pretty well in the seedlings at about the 40 micromolar level of selenium. And then over here, this again is showing the uptake in the tissue concentration of selenium. So this 80 and 160 micromolar did pretty good. And the red is the roots, the red is the shoot, and the black is the root. And then down here, this is in mature. And they did real well. So like right here would be 10 micromolar of selenium. And you can see that the plant biomass was up around 8 grams versus this one, which was around 80 milligrams. And then this one this is immature again, and this shows all the different organs and how well they did. So again, we have selenium on the bottom with the tissue concentration. And the green is leaf, and the red is flower, and the black right here is, or the dark is the seed, and then the black over here is root, and the blue is stem. So it did mostly uptake into the leaf and into the flower, and a bit into the seed as well. So it did pretty good. So hemp definitely looks like it can do some phytoremediating. So my research questions are, will cannabinoids be affected by these inorganic pollutants? And will the cannabinoids be harvestable for other uses after the pollutants are taken up? So my objectives are to understand how cannabinoids are affected when hemp is grown with different inorganics, and to discover if the cannabinoids will be contaminated with pollutants. So the goals are to treat hemp with different inorganic elements, measure the cannabinoid levels, and analyze the compounds for the presence of pollutants. So my experimental plan is to grow hemp to maturity using feminized seed in a greenhouse, something like this, to grow sunflowers as a control. And I would treat it with a number of different elements, arsenic, cobalt, mercury, manganese, molybdenum, vanadium, and tungsten, just to look at some different inorganics and see how well it does. I would do 5 replicates, 4 different concentrations for each one, so that's going to be 20 plants per element. After growing, then we would harvest, dry, and separate the flowers, stems, leaves, seeds, and roots. So we want to look at all the different organs to see where the hemp's going, where the pollutant is going, as well as to check out the trichomes. So we're going to extract the compounds from the trichomes, and we'll analyze them using gas chromatography, and we're specifically looking at THC, CBD, CBC, and CBN, and then to analyze the extracts for contamination using ICP-OES. So my methods would be for extraction. There's 2 ways to do this. So ethanol or CO2 extraction. So essentially you want to dry the material and ground it down into a powder. And then you would add ethanol. And ethanol is safer than other solvents. It does not have any residues. And ethanol is polar, so it'll attract the water. However, it's also going to attract the chlorophyll, which isn't the greatest. But then you soak it in the ethanol, and that's going to separate your compounds out of the hemp tissue, and then filter and concentrate it by removing the ethanol and drying it. And then the only problem, though, is that this also is going to pull out the plant waxes. So the CO2 extraction is actually better. It uses supercooled CO2. It does essentially the same way that this one did it, except it's safer, you don't get any tissue damage, and you get to recapture your CO2, which gives you a more pure extract. And this is a machine of a CO2 extraction machine. And so then the second thing I would do after extracting would be gas chromatography. So this screens, identifies, and quantifies nonpolar compounds. So now we're going to find out which different cannabinoids we have. So it separates based on boiling point and vaporizing point and polarity. So you take your samples and you put them right here in this little injection port, and then as it's coming down, it's met with a carrier gas. This is typically either nitrogen, helium, or hydrogen, but from what I read, helium is used the most. And the helium moves the sample down into this column, which is sort of like a drying oven, but it goes around and around here. And as the heat is going around, it's actually separating it. Remember, it's based on its boiling point and the vaporization point. So as it gets separated, then it comes out here, and these components pass through the detector. So they have been separated out, and then there's an electric signal that's created that's proportional to the amount of the analyte that's there. And then that's captured on the computer. So when you get a readout, you're going to get peaks, and each peak would identify a different compound. And then on your y-axis, you'd have the abundance or intensity of that peak, and versus the time on the x-axis. And then the third thing would be to look at the extract to see if it is containing any of the toxins that we originally tried to phytoremediate. So this would be done through ICP-OES, which is inductively coupled plasma optical emission spectroscopy. And you have a sample, you digest it with nitric acid, and then it's taken to this machine, and you kind of sit them in this tray that sits right here next to the machine. And there's an arm that sticks up right here, and it plops into each of the little tubes, and it sucks the material it's just a liquid now, into the machine, and it sort of nebulizes it. So it sprays it, and then you get a pattern which is identifiable to certain elements. And so it can tell you what elements are there, and it also tells you the quantity, how much is there. And then you just compare this with what you know about your sample. So whether it was a leaf or whether it was a stem, and you know then where the fate of that element was, where did it go into the plant. Okay, so for discussion of the knowledge that would be gained by this research would be to identify what compounds are present in hemp flowers and their different organs, and as well as the fate of those contaminants. We would determine if the compounds are affected by inorganic uptake of the hemp, and determine if any of the pollutants are in the compound. And the significance of this research is to better able to use hemp for phytoremediating, so of inorganics, sorry. So it would help in choosing the potential inorganics, choosing the appropriate phytotechnology to use. And then with post-harvest recovery of cannabinoids, we would be able to possibly capture the economic value of, and this may be more than just the cannabinoids that might be available for other things. It could be other parts of the plant, like if the excuse me, if the toxins only went into the roots, then you could capture the economic value of the stems and shoots as part as well. So possible further research would be looking at evaluating other cannabinoids. There's over 100 of them, and many of them I don't think have been looked at very much, as well as all the terpenoids. And there's over 140 of those so far that we know of. Also, we could look further at other factors that might be affecting cannabinoids, like, what are environmental conditions doing, and how do they affect the cannabinoids that are present or that are created and synthesized, and how much is created. And we also could look at the other pathways for these other compounds to see if there's other ways that we could be controlling things, especially when we're looking at something like the THC versus the CBD levels. Okay, so my question for everybody is, where are the cannabinoids and terpenoids synthesized in hemp? And that is it. Here's my sources. And thank you everyone for listening.